Is it recording? Okay, it's recording. Well, hi everyone. Thank you guys so much for coming today. This is our second GBM of the quarter, and we are the Youth Movement Against Alzheimer's. We are a chapter of a larger nonprofit called the Youth Movement Against Alzheimer's, and we're completely youth-led, and we focus on promoting understanding of Alzheimer's in the youth and young adults and providing opportunities to help those affected by this disease. And that includes caregivers, families, and the individual themselves. And this is all through some fundraising, advocacy, and service events. So thank you guys for coming today, and we'll show you guys what we have for you guys this quarter.
Hi. So we have a few volunteer opportunities that you can take part in. So first off, we have the Phone Pals program, which we're starting this quarter. This is a program where you connect volunteers with the elders, and you're just going to help them feel less isolated. You're going to have to call them once or twice a week for just at least a few minutes, or you can just do more if you want. There's also the Valentine cards, where it's just going to be virtual Valentine cards you're going to make for elders. And there's an opportunity with Elder Help, and the position at Elder Help is just you're going to help elders drive them to medical appointments and just help them with essentials and food and stuff. And then there's also a position with the Alzheimer's Disease Research Center where you're just going to sit and brain cutting and stuff like that. Pre-COVID, there was a lot more to do, but right now, obviously, the situation, you can't really do much, but it's still a really good opportunity.
Hi everyone. I'm going to talk about our fundraising opportunities. So the first one I want to mention is our 135 fundraising campaign, and what that is our the entire WIMA, the national board, and all its chapters just launched our 135 campaign to support chapter advocacy and caregiver respite programs. And how it works is you would pledge a small monthly donation, like $1, $3, or $5, or you could just do a one-time donation. So we're going to post the link to that on our social media pages, which you can find later in the slides. And then I know Nancy touched on this a little bit. We're doing a Valentine's Day fundraiser to give cards to the elderly, and we're doing this in partnership with Undies for Oldies, another elderly help service organization at UCSD. And with $1 donations, you can send a gram to seniors at serving seniors and other senior care facilities throughout San Diego. And then I'm gonna talk more on the next slide about our sticker fundraiser. So we are finally launching our sticker fundraiser, and this will fund a lot of Alzheimer's, we will donate the money towards Alzheimer's research and our many volunteer opportunities and events. And then in the next page, these are all the sticker designs we have right now to purchase. So if you're interested, stickers run for $1, and then there's a custom sticker that you can get for $150, and I will design you as a little in that art form. And yeah, and it's free shipping. It's a good small gift for Valentine's Day, because that's coming up. Or you can, if you don't have anyone to buy them for, you can buy it for yourself, because self-love is very important. And yeah, so stickers will be 2.5 times 2.5 inches and a glossy and weatherproof finish. Alrighty, so here's the link to the sticker order form, and we're going to close orders after 10 orders just so I can process it, and because we don't want too many people touching the stickers, especially now, and then reopen it once we're done processing those orders. So order now before it's too late. And then we'll have this QR code and the form link at the end of the slides, so you can take your time and look that up.
Okay, so for the main courses tonight, we have our neuroscience research talk with Edwin, specifically Neptrin 1 signaling in Alzheimer's disease. So Edwin is a medical student at the University of Toronto right now, and he's also a passionate neuroscience researcher and educator. So yeah, once Edwin is done with his presentation, we'll have a Q&A session. So please hold your questions till the end. Thank you very much. And Edwin, the floor is all yours.
Thank you so much for the introduction. First of all, very cute stickers. I love the initiatives, way more than what I would have been able to do. Mostly the fact that like if I draw them myself, this patient would probably be a little sicker. Okay, so first of all, I just want to thank Wei Wei for inviting me. She came to a talk I gave in a conference in the summertime, and I'm very happy that it was relatively well received and was good enough that she wanted to invite me and give a talk to you all. So as with the introduction, I am currently a medical student at the University of Toronto. For those who don't know, that means I'm in Canada or Canadian. And I've done a master's degree in neuroscience, and I'll touch a little bit about my research and academic career, as I think many of you are interested in pursuing either research or maybe in medicine. So I've been told to maybe give a little bit about my journey as well. So I'm going to do a bit of a icebreaker for you all. So I'll probably need you to kind of get your phones. It's not going to be super cheesy. I'm not going to ask you to give like a not dumb, sorry, bad word, like a cheesy, like fun fact, but if you can go to the website, menteecom, right here, I'm gonna ask you just a couple of questions just so that I can get the vibe of what the group is kind of like, especially when I tailor it to the talk. Okay, so it's going to, so if you can go to the top here, mentee.com, there should be a place where you can include the code. So the code is 1029396. And whenever you are ready, you can answer the first question I have for you all. So what is your year of study and what are you interested in pursuing after undergrad? You can also include your program as well. It'd be kind of nice to see if you're in humanities or more in the basic life sciences. That would be also good to know. And let me know if you are having any difficulties with that. Ooh, okay. So we have fourth year biochemistry, want to go to neurosurge, ambitious, I love it. Some of you are interested in going to PA school, some of you are interested in maybe multiple healthcare like PA, MD, or nursing. Dentistry is great. A couple of them are into research, some of them are not sure yet. That is fantastic, I love it. And one of you is planning to do a PhD, MD. And OT, that's a fantastic field as well. So this is great. Usually I give a plug in the like, you know, a lot of the keeners and med school pre-gunners are like, I want to go into med school, but they also don't know that there's multiple types of healthcare professions. So it's fantastic that you are already aware of that. Next question. Oops, that's not what I want. Next question. What interests you about Alzheimer's disease or neuroscience in general? So I just want to give you couple of minutes for that. And it seems like a great organization. It's not like a super like only research, but you're also doing some really nice initiatives. So that's really great to see as well, more of like a practical aspect versus just like a journal club, boring, or not really boring, but like science. Aging of the brain, dealing with the older population, so geriatrics, that's fantastic. I just rhymed there. Memory, Okay, yeah, so memory and synaptic plasticity, if some of you may have heard about that term, very hot topic, something that I've been personally interested in myself as a researcher. Maybe a few more minutes just for some last minute tricklers. And some of you are maybe thinking about it, and you don't know, and it's just neuroscience is a great thing to do. That's awesome as well. Find how complex the disease is. Absolutely, Alzheimer's, I find it's one of those diseases where everyone studies it because it's such a hot topic, but no one really understands why. Memory aging, fantastic. So it's a huge prevalence, especially in North America. So that's great. So I'm gonna quickly move on to that's my paper and that's my study notes. Don't look at that. Okay, so I'm just gonna quickly change my view. Fantastic, okay. So those are some great ideas and answers. Thank you guys for participating. So a little brief intro about myself. I went to McMaster University, so that's in Hamilton, Ontario. I did a bachelor's in science in biology and psychology with a minor in math. So for those who are aware of the term, that's very Asian of me to do that. This is probably me in first year being a dweeb, so nothing has really changed on Halloween. And this is me during my graduation ceremony for my graduates. So it's very cute, very kawaii. Is my mom and dad. I'm not Japanese, by the way. And it's when I celebrated my graduation. And finally, this is me. I'm currently a second-year medical student at the University of Toronto. So next year I'll be in clerkship. For those who don't know what that means, clerkship just means that I'll be spending almost all my time within the hospital for the next couple of years. And by next couple of years, I mean forever in my life. So what took me so long to decide to apply? So a little bit about my background. I did not ever want to go into medical school during undergrad or even throughout most of my master's. A lot of the reason is because a combination, for example, pre-med toxicity, some of you maybe are already experiencing that, where some of your peers are kind of comparing back and forth, especially doing the MCAT, doing a million hundred things to get their application and CV ready. And I didn't feel like that was for me. I'm a competitive person, but I'm also very intimidated by people who are very big personalities and want to do everything. Parental pressure. So of course, coming from that Asian stereotype, I came from a very traditional Asian household. So success was defined as either being a lawyer, doctor, or engineer. And if you don't fit that criteria, unfortunately, that may be looked down upon. So for me, it was almost like a rebellion. So I didn't want to go into medicine or those fields because my parent or other family members told me so. I wanted to do it because I did it myself and I wanted for that. And it took me a long time to figure that out. And career exploration. So this is me in the lab. Again, I didn't want to go into medical school early on, and I really wanted to be a faculty member. I wanted to do teaching, still want to do teaching, but I also wanted to be working at a university. So doing a master's and potentially a PhD was something I was very interested in. And that was kind of the path that I took. Obviously, nothing is as straightforward as it sounds in life, and you just have to embrace all those new challenges and all those new changes. And that's what makes life so exciting. And finally, the fear of failing. So it took me a long time to realize maybe a large part of why I didn't want to go into medicine is because it's so competitive. Not getting in maybe is a sign of me as a person, me as a success or a failure. And that is something that I put myself as a label. And I'm sure many of you have very high standards for yourselves. And what I say to that now, going through so many years of thinking and figuring out what I want to do with my life, is that failing is sometimes the most beautiful and amazing thing you can do. As cheesy as it sounds, I failed so many times, and you can only learn more from it. And what is life so exciting if you're going to be always getting what you want and being successful in every single thing you do? And success is different for every single person as well. So as the introduction, I forgot who gave, I think it's Wavy or someone, I forgot, apologies. But they did mention that I was very heavily interested in teaching and education. So during my gap year of my master's and applying to medicine, I was actually a high school teacher. And I was also currently as a lecturer at McGill. So this is me teaching in a different initiative and me giving a lecture to undergrads like yourselves. So for me, it was a great, fantastic time to take a gap year to really do things outside that I wouldn't have been able to if I went straightly into a particular career. It really improved my communication skills, but furthermore, and most importantly, it furthered my passion for teaching. It's something I'm still doing till this day. I'm planning to maybe do like a medical education degree afterwards to do more teaching outside of medicine, and something that I may not have been able to realize outside of this gap year. So, you know, taking some time and taking it slowly can also be an amazing opportunity as well. So enough of that cheesy talk, time for what you actually are interested in, and it is my research and what I have been kind of procrastinating or not doing as much in these pictures. So as the title has mentioned, I work on a protein or worked on a protein called Neptrin-1. And Neptrin-1 has a primary receptor called deleted in colorectal cancer, or DCC for short. Their awful name, biologists have the worst naming system. It was found in colorectal cancer, but then now it's discovered in the brain. So now it's too late to change the name while making it super awkward. So they just kept it as DCC. But essentially, these two proteins are very important for the development of the nervous system. This was spearheaded by my supervisor in McGill, Dr. Timothy Kennedy. He was actually a huge person in the field where he discovered Neptrin-1 was in fact really important for the development of the nervous system. So how is it expressed and how is it really working? So this is a kind of a picture, a schematic of a embryonic spinal cord. I believe this is a chick, so like an animal bird chick, not like a person chick. So as you can see here, the green represents the concentration gradients of Neptrin-1. It is kind of expressed very highly in the very bottom and slowly decreasing its concentration as you reach the top of the spinal cord. This is really important because the different concentration actually allows different signaling pathway. The more the concentration, the more certain cells will be like, ah, I want to go towards this. So this process is actually called chemoattraction. So certain molecules would be very high in abundance, and it's acting as like a signaling flag, like, hey, cells, come towards me. This is really important because, as you can imagine, the brain is a two hemispheres. So you need to have cells going from one other hemisphere crossing to the other hemisphere. Structures like the corpus callosum, hippocampus, which are bilateral structures, will require this information, or else only half of it will be formed and the other half will be absent. And this is where, you know, things like fetal development, impairment, all that kind of stuff will occur if you don't have this proper signaling. Just to kind of mention that Neptrin-1 has multiple receptors, but the main one we'll be talking about is DCC. And this is just like a cute little image that you can stain Neptrin-1 in the spinal cord. So this kind of attraction towards cells attracting to the Neptrin gradient is called Axon Guidance. So the axons are physically reaching, and I always say that videos are way more important than what I can ever say. So here is an image or a video of a cell undergoing axon guidance. You can see that the axons are actually protruding. It's almost sensing its environment as it's growing. So millions and billions of these are constantly doing that during development. And you know, you can do a lot of fun things with this. Neptrin is such a strong attractant that you can actually lay it on like a little pattern, and you can see the axons grow in the pattern that you want it to. My lab has been, you know, been kind of a funny lab, and they do like little paintings with the axons depending on where you kind of drop the little Neptrin proteins across the sheet. So this is really great, but my project is focusing on how Neptrin and DC are important in the adult brain. Everything I've been showing is in the embryonic developmental stage. So we know that Neptrin and DC are heavily expressed throughout aging, throughout growing up. So what are they doing there in the brain? So to answer this, you kind of have to revisit the amazing favorite brain structure in my life, the hippocampus, which I'm sure many of you are, especially with Alzheimer's interest. So just to quickly reiterate, the hippocampus is like a seahorse in Greek because it looks like a seahorse for some reason. There are three main structures or components of the hippocampus. This is the CA1 in the light green. The dark bluish green is the CA3, and then the blue is the dentate gyrus. Together, they form what we call the trisynaptic circuit, really, really important in things like LTP or long-term potentiation or synaptic plasticity, which is kind of the basis for molecular memory formation. So how does Neptrin and DCC kind of play into this? So we found that DCC is really, really important in spatial memory and regulating synaptic plasticity in the brain. In our lab, we have found that DCC are heavily expressed in these little punctates. So this is actually a axon. And then, and you have these little spines here. See it? These spines are called dendritic spines. So dendritic spines are actually thought to be potential memory markers that hold information that's really important for the formation of memory. So this is kind of like the actual, like you can touch and feel a memory. They found that DCC are heavily expressed on these spines. And if you actually delete DCC in animals, so here is actually a deletion of DCC animals, and you do electrophysiology experiments, you find that there is an absence of potentiation. So you remove synaptic plasticity when you delete DCC in the forebrain, which is pretty cool. And of course, you want to see whether or not it is important when you look at behavior. So Morris water maze is a very common spatial memory test for rodents. So for those who are unaware, it is basically a large water pool, and underneath is a platform. And what you do is that you gently place the animal into this water pool, and they need to find this little platform hidden underneath. It's also very cold, so as you can imagine, the animals are not super keen and happy about this. But over time, you do this for a couple of days to about a week, they should be very, very good at finding this platform. Well, on the last day, because we are sadists and we're scientists, we actually remove the platform, and we can actually measure with a camera on the top how much time and distance they spend in the target quadrant if you divide the pool into 4s. So the more intact the memory, the better the memory, the more time and distance they would spend in the target quadrant. The poorer the memory, then the animal probably has nowhere to go, or they don't know where to go, and they will spend equal amount of time in all 4 quadrants, if that makes sense. So again, camera's video is way better. So here is a person placing a rat into the Morris water maze. And you can see here, this little circle is the hidden platform. So you're like, oh no, you're almost there. Oh wait, oh no, you missed it. And then he's like, oh, or she, I don't know what the sex is. There it is, found it. So as you can imagine, the more you do it, the better the animal are at this test. So as you can imagine, because with the way I'm giving this talk, deletion of DCC actually impairs the animals in this particular test. But not just in any animals, actually older animals, which is kind of in relation to the Alzheimer's aspect I'm going to talk a little bit about in a few slides. So only the older animals show impairments, whereas, or at least significant impairments, whereas the younger animals do not. So if you want to give in context, rodents usually live about 2 years or 24 months. So 8 to 10 months is actually about middle age. 2 to 4 months is about their angsty teenage phase. And what I did when I came into the lab is that I wanted to further investigate this phenomenon. So not only did I delete DCC, but I only deleted DCC in either the CA1 or just in the CA3 to see if this is actually a selective phenomenon or not, or just like a full deletion of DCC. So again, CA1 is right here, CA3 is here, dentate gyrus is right here. So I had 2 lines of mice, one deleted here and one deleted here using genetic knockouts. And what I did is I did a different test. This is actually called a novel location object test. So you give the animal, this is really cute, I actually did this myself for the paper. I have the animal, I give them about 5 minutes to investigate 2 identical objects. So they're now aware of these objects. I remove the animal for about 2 hours, or sorry, 24 hours. And then the next day, I actually put them back in the exact same box. The only difference is that I now moved one of the objects to a different area of the box, okay? So now this is a spatial test because the only difference is the location of the object. So, and animals love novelty. So if they have intact spatial memory, they should actually recognize that this will be the newer, newer object, and they would investigate it more. If they had no sense of spatial memory, they would investigate both of these objects relatively the same. So the red graphs are the CA1 deletion. So as you can notice that there's a significant amount of difference compared to controls. So the animals actually do not investigate the new moved object compared to controls. And when I deleted the animals, you see in the CA3 region only, in the green, similar effect. Now there is also a selective phenotype with this deletion. So not just DC full out in the forebrain, but also in selective hippocampal regions, which is really, really fascinating. And that was something we were able to publish last year. So kind of now moving from DC to Neptrin. So Neptrin is the ligand. We also show that they have very similar phenomenon. So for electrophysiology peeps in the group here, if you just take normal neurons and you give Neptrin and do nothing to it, they actually will spontaneously elicit LTP. So this is fantastic. It's actually quite amazing because you just don't do anything, right? And you actually now have synaptic plasticity in front of your eyes. And it's also a dose-dependent effect. So the more Neptrin you give, the higher the potentiation. So now we have two conclusions from these electrophysiology experiments. Endogenous Neptrin-1 in the hippocampus is required for synaptic potentiation. And the second, if you give exogenous Neptrin-1, so just give outside Neptrin-1, it is sufficient, right? So if you're in biology classes, those necessary versus sufficient, those are what is really important now in science. Is it necessary or sufficient, or necessary and sufficient, right? Kills me every time doing those tests. So moving on to Neptrin-1, again, those dendritic spines that I mentioned a little bit before, if you add Neptrin-1, you can actually see in real time, they change shape. So I wanna focus you on this. So that blue dot means that we just added Neptrin. And you see this little spine here that we focused is actually getting a little bigger in real time. And this is by hour. So it's about an hour and 18 minutes. So again, I'm gonna show you one more time. So it's kind of like a little lanky, doing nothing. And then once you, boom, add Neptrin, you start to see it actually increasing. It's not a lot, but as you can imagine, the dendritic spines are not that big to begin with. So moving just a little bit actually demonstrates a huge effect synaptically. And for those who are still questioning what the hell did I just watch and was there even a difference or was it just like the video being crappy? I will show you, if you take screenshots of it, now there is an actual physical difference that you can actually measure using a ruler. So that's molecular electrophysiology experiments. Now we move on to the behavioral, and this is very similar to what a lot of scientists do. They do it in the micro aspect, and then they move it to the macro aspect and see if there's a behavioral change now. And for me, I never really like electrophysiology, so most of my work in the lab was more behavioral. That's something that I specialized in the lab. So again, I took two groups of animals, one control or wild type, so it did nothing to it. And the other one, I deleted Neptrin throughout the forebrain. For those who don't know, the forebrain is the large part of the frontal lobe that includes the hippocampus. Not the frontal lobe, the frontal hemisphere as well. So if you were to do the Morse water maze, so this is actually the experiment and paper that I published in the lab. If you actually take the Neptrin-1 deleted mice, put them into the spatial memory test using the Morse water maze, you find that they do not cross the platform as many times as control mice. And if you look at the time spent in the target quadrant, so I want you to focus only on this graph, they don't spend that much time in the target quadrant, as well as the distance as well. So now you see that there's a receptor aspect as well as a ligand aspect. That's really important. And that's actually kind of rare sometimes to really see this phenomenon where you both see a similar aspect or a result from deleting just the receptor or also just deleting the ligand. So what does this all kind of mean to you and how does it relate to Alzheimer's? Well, there has been increasing evidence to really show that Neptrin-1 might be involved in Alzheimer's pathology. There is growing evidence to show that it interacts with a protein called amyloid precursor protein, which I'll go into in a little bit. And more and more research over the past couple of years are growing that Neptrin-1 may be implicated into this research. So when you actually group all the different studies together, the most common Alzheimer's related protein that Neptrin-1 seems to be focused on is amyloid precursor protein, or APP for short. So what is this APP protein? Some of you may have heard about it, but just to cover all grounds, I'm going to quickly give a little lesson on it. So APP is involved in a hypothesis many Alzheimer scientists believe is involved in this pathology called the amyloid cascade hypothesis. So here is a lipid bilayer of the cell membrane. APP is a transmembrane protein, so it means that it actually inserts throughout the lipid bilayer. So this protein tends to be cut a lot by different enzymes. Depending on who cuts it, it can go into either the amylogenic pathway or the non-amylogenic pathway. And for those visual learners, the red just means bad and green means good, for simplicity's sake. So let's move on to the good side first, okay? If you get cut by the alpha secretase enzyme on the left side here, it will produce a fragment which is then further cleaved by another enzyme called gamma secretase. And it produces a protein called P3. Okay, P3 is a very non-toxic protein, has some beneficial functions physiologically in the body as well. And I forgot to orient you all, that's my mistake. This is the extracellular part of the cell, and this is the intracellular part of the cell. So all the enzymes are cutting it in the extracellular aspect, minus this guy, who's kind of like intercellular. So if you go on to the bad side, so the red side now, if you get cleaved by beta secretase first, you produce another fragment, which is also cleaved by gamma secretase, interestingly enough. So this guy pops in twice. But you don't get the P3 aspect now. You actually get an A beta fragment. So for those who know about the Alzheimer's pathology, A beta is actually the fragment proteins that end up clumping together extracellularly, producing these A beta plaques, which is one of the hallmarks of Alzheimer's pathology. So this hypothesis has been investigated so many times for so many years that there have been treatments developed to kind of counteract this phenomenon on the right side of the pathway. So for example, certain drugs have been developed to impair or inactivate beta secretase. Some of them have been inactivated gamma secretase. Some of them are actually increasing the activity of alpha secretase. Sounds kind of interesting because if you activate alpha secretase, it might actually tip the balance now. So you're having more activity on the left side and reducing activity on the right side. So there has been growing evidence to suggest that Neptrin-1 might be a ligand for APP. So meaning that Neptrin-1 might be floating somewhere here, it might bind to APP and somehow having some unknown effects. So that's really interesting to see what these effects are. So kind of finishing up, because I might be exceeding my time limit here. What is next for me? Hopefully you're still awake for listening, but I really want to continue on my career, combining a little bit of teaching, a little bit of clinical aspect, and a little bit of research. For those who are interested to know, I'm really interested in maybe going to neurology, which kind of makes sense because the brain and stuff like that. But I'm open-minded, and that's something that you would like to think about when you're going to medicine. Not just going spearheaded towards one field, but really opening your eyes and your ears to other aspects of medicine as well. Teaching is a huge thing for me. I don't think I could live without teaching anymore. It's something that I do therapeutically for me. It is a way to de-stress. So this is a way for me to strangely de-stress as well. So if you have any questions, feel free to contact me at any time. I welcome any questions. Again, thank you for having me. And yeah.
Thank you so much, Edwin. That was amazing. And I died at your jokes. You're so funny. Oh my God. And so, yeah, if any of you have any questions, feel free to drop them in the chat or just unmute yourself.
Could
Yes.
you type sorry,
Sure.
could you type your email in the
Alright.
chat?
Sure. I'm glad everyone likes my jokes because they're pretty dad jokes. And sometimes I make inappropriate jokes. I'm like, ooh, can't say that.
Not so much about research, but I'm interested in how you taught in your gap year and how you got into that and how you actually were able to teach at a high school.
Yeah, so that's a great question. So I've been teaching for a number of years as a teaching assistant during undergrad as well as grad school. And I was actually really fortunate that some of the professors I work with when I TA'd allowed me to give guest lectures, which ended up being more of a consistent thing. So I had some teaching experience under my belt. During my gap year, I still didn't really know what to do in terms of research. I want to go into medicine and make my parents proud, you know, yada, yada. And I actually was applying to clinical psychology programs for a couple of years. So I didn't end up getting into those programs. They're quite competitive. So I ended up applying to medicine on my third try. I was like a backup. Sorry. Yeah, sounds like a little, it's not a backup plan, it's just a change in trajectory. But anyways, to answer your question, during my gap year, I was working part-time in the lab, finishing up some projects, and I was working as a private tutor with a tutoring company in Montreal, where I studied. So in Montreal, very interestingly, high school teachers do not require a teaching certification or an extreme certification that you may need in other provinces or in the States, as long as you have a master's or a PhD in the subject that you're teaching. So I was very fortunate that one of the schools that were partnering with the tutoring company, they fired one of their science teachers, and then the company recommended me to take over. It was an all-girls school. It was really sweet. It was a grade 10 science class, and I loved the students there, and that was something that I continued doing on for the rest of the year. So really enjoyed it. Watched a lot of TV, watch a little bit of RuPaul here and there, watch drama in the middle of like lessons. So it was great.
Yeah, that's really cool. I actually like want to go into like being a physician and being like a professor as well in
Fantastic.
medicine. So
Yeah, no, absolutely. I'm very interested in like the combination of teaching. It's something that I think it's not emphasized enough in medicine, because everyone's like, oh, you need to get a medical education, but then you're like, teaching is not a priority for students to learn how to teach. And that's something that's kind of contradictory in my head. I've actually started a couple of teaching initiatives within my medical school, where now medical students in my class are able to now teach. That's something that if you're interested in doing, I think I gave away some of the information. Every 2 weeks, medical students from my class will give a lecture to the public on a medical topic that they're interested in. So if you want, you can just come and join and listen and stuff.
yeah, I think Wei mentioned that to us before, so we'll post about that on our like social media just to have people like come out, check you guys out. And
Yeah.
I can tell like the teaching really, like teaching all those years really paid off because today's presentation was really good.
Oh, thank you. Very sweet. Thank you. I like put it last minute together. I'm like, I don't know if this is good because I haven't like seen some of these slides in a million years. Hopefully it wasn't too advanced. I didn't know what the level is. It's definitely a stretch from high school presentations that I usually give before. So I was like, hopefully it's like easy enough to understand.
Yeah, no, it was really good. Like, thank you for all the animations and videos and visuals. Definitely learned better with those than just like words. But yeah, thank you so much. And I just have another I have a question. So being a medical student, right? Like, how do you feel? How is it right now? Is it as scary as they say? Like, you memorize a book in a week?
Yeah, no, so medical school, again, there's a lot of like these stereotypes which you will never know until you actually get into it. And then you laugh and you're like, we just cry a little less. I'm joking. But there are sometimes tears. I think medical school, obviously, when you go into it, it is not meant to be easy. If it is easy, then, you know, why would there be so much competition to begin with, right? And why is it so rigorous? Because lives are at stake, obviously. So there is an enormous amount of information to intake, especially when you're going from like cardiology, then suddenly go into respirology in like a few weeks, right? And for your undergrads, you're taking a whole course, but they're spread across 4 months. We would learn maybe a course or a subject very in depth within 2 weeks. So the very famous Lowe phrase is that you're almost drinking from a fire hydrant because there's a lot of information. That's not to say that it's impossible, because again, I'm not the brightest person here, so obviously I'm still surviving. That means a lot. Medical school and the medicine that you learned are something that you will see over and over and over again. I think we're so trained as type A personalities that want to get everything done perfectly the first time. We're so keen on being perfect. But the reality is medicine's not perfect. It's going to be so many different circumstances. You see a heart attack, for example, right? And the knowledge that you gain, you'll see it in not just one time, you'll see it so many times to the point where it will be in the back of your hands at the end of the day. So for me and some of my friends that are now kind of mentoring incoming medical students, we're like, holy crap, we actually learned so much. The amount of material that we've learned and digested and then still remember is actually quite amazing. From being a baby medical student last year, where we're like, I don't know how to wear this thing, but like, you know, so it's not too bad. And I think it's something that's very great field if you're very passionate about.
Thank you so much for sharing.
Yeah, no worries.
I also had a question. This was related to Neptrin. So you know how it promotes spine growth? Then if you take it away, does the newly formed spines die, or does it stay alive?
Yeah, that's a good question. There have been some studies that the postdoc that I worked really closely on a number of the papers that I presented today, he actually kind of looked at longevity and how long there's an effect of Neptrin-1. So the spines don't die necessarily, but they may regress back into like a smaller form, right? So dendritic spines come into kind of 3 different flavors. They come in mushroom spines, come in thin spine, and they come in like spiny spine, thin, stubby, and I forgot the last one for the life of me. But essentially, the different configurations of the spine just talk about how stable they are. For example, mushroom spine are the most stable, meaning that if you have those, you can kind of equate to like long-term memory because they're the most stable type of memory. Versus thin spines are very transient. So they come here, pop, and then they go back into like a little stubby spine. That's what's called, stubby spines. Anyways, so then thin spines are very transient. So they're kind of like short-term memory. They come and go, right? You hear one thing and you forget the next day. So Neptrin-1 has quite a long, it also depends on how long you leave Neptrin in for. You also don't want to be exposing the cells for, well, nothing for too long. And I'm not entirely sure what the exact length is because I'm not a big electrophysiology person, but they don't die necessarily. They just change back.
Okay, thank you.
Yeah, no worries.
And then I had another question. So in your last slide, you said you want to do research. Then like, did you ever consider like getting an MD PhD? Because I know you did your master's and I don't know.
Yeah, no, it's a fantastic question. So yeah, I think 1 or 2 people in the earlier question that gave or interested in maybe doing an MD PhD. So that has been something I thought about. My supervisor actually highly encouraged me to do an MD PhD, to stay in this lab and then just do medicine on the side. Reason why I didn't want to do an MD PhD is very simple. I had enough of research for a little while. If you count, so I did a 3-year master's program. So traditionally masters are about 2 years, but I was very fortunate that I got a scholarship near the end of my 2 years. And because I didn't get into clinical psych, that was kind of an opportunity, like a silver lining, to stay in the lab, to finish up some papers, and well as get some publications and some extra money. And then I ended up staying for a 4th year as a research assistant, because I was taking that gap year, right? So for me, that was like 4 years me in the lab. if you add another PhD, that's about 4 to 5 years. And I was like, I'm okay with research for a while. I'm doing research still, but it's very light and something that's very different from Neptrin. And I wanted to focus on just medicine.
Okay.
And if you want me to go into depth a little bit later in the discussion, the MD PhD program is a little different. You don't do just, you do like MD for like 1 or 2 years, and then you do a PhD afterwards for like 4 or 5 years straight. So I didn't want to forget all the medicine as well.
Okay, that makes sense. Thank you.
Yeah, no worries. Any other questions?
What other extracurriculars are you currently involved in outside of your light research?
Yeah. So kind of answering I think Wavy's question originally, like, is it scary sounds? No, because there's a lot of extracurriculars. That'll be very dependent on the school that you go into. For me, I was very interested in music. I played piano for a number of years, but I love singing, but like singing in the shower singing. And U of T actually holds an annual musical show that's produced, written by medical students for Cancer Society. And I tried out, and I actually got a part to be a cast member. So I had to sing, dance, and play, and then act on stage live. And that's a tradition that our school has been doing for about 130 years. So that's something that was very cool to actually do, like a Broadway show in the middle of studying medicine. Other than that, I've been doing a lot of teaching initiatives this past year. So I've been teaching a lot of like youth. So I'm organizing a neuroscience course for underprivileged high school students that I'm actually spearheading myself, and teaching it throughout Canada. Thank you. So I'm very excited. I'll be starting in a couple of weeks. And I've been doing a lot of like outreach, so helping underprivileged students, specifically Black and Aboriginal students that want to go into medicine, but maybe not have the opportunity to do. I'm partnering with a couple of organizations to maybe help read the applications, do webinars of like tricks and tips to get into medical school and research overall. And I'm teaching a bunch of things. So I'm also teaching a stats program for those who are interested in research. So yeah, I'm doing a lot.
Yeah, I always like asking medical students like what they're doing outside of medical school, and yeah, it's always refreshing to hear, just because like it's so dreadful to say like, oh, you're just studying a lot.
Yeah, and and the thing is, like, studying is so different for each person. I'm a big studier. I actually like studying. I like organizing my notes. I like going through everything at least a couple of times. I'm not a gifted person where I can see something and memorize it really quickly. I have a really good friend. She is a genius. She's not here, so I can say it. She scored 100 percentile in the MCAT, and she will study maybe the night before the exam and still do better than everyone else in the class. And she's just like, oh, I don't know, I just guessed it. I'm like, you guess every test. So you have people like that. And then she just like paints on the side, and she does a lot of like exercise and yoga. And I'm like, how the hell do you have time to do all this? So it's very variety, right? And it's actually really nice to show that doctors and medical students are not robots, because we're not humans. We like to cry. We like to like go out and do things, and you should, because you shouldn't be a bookworm, because that's boring as hell.
Yeah, it sounds fun.
Yeah, I mean, you have to kind of make the best of your time, and you have to prioritize. I find that a lot of it has to deal with age gap. I'm more of a mature student entering medicine versus some of my friends and colleagues that enter right after 3 or 4 years of undergrad only. So priorities are a little different, and for me, I know what I need to do to make myself happy, and younger students of mine or younger colleagues of mine may have like, I want to do certain specialties, and that's my one goal. And for me, I'm like, I've worked my ass off for so many years. Why do I want to stress myself with just one specialty and just enjoy other things as well? So, you know, to each their own.
Yeah.
You're so cool. Like I just have to say it, like, you like know what you want, like you said, and then like we can like totally just see your passion for it. And it's just like, it's amazing. I feel like a lot of people are jaded by now, but it's good to see.
Oh, thank you. So it's funny. Two of my best friends in medical school, we call ourselves the Three Musketeers. We're very cynical, jaded people. But like in a way, we don't take ourselves too seriously. In a way, we're like, oh, why are people like so stressed about this? Like I'm stressed enough. I don't need other people to stress me out, right? So I think I learned to filter negativity out as much as I can. People are competitive no matter what stage you are in life. And I'm glad I give the persona to have my shit together, but I was recording, have my life together.
I can cut it out.
But, you know, I have a lot of insecurities. I have a lot of doubts, and it all ends up being a support system and making sure that you have people that you care and love about, that you trust. That's really, really important, especially during COVID season and during your undergrad studies. It's really important to develop a very strong social group, friends and family likewise, to really support you. And that's all you can really ask for in life.
Wow. I'm just like, cool.
So inspiring.
Thank you. Thank you.
Yeah, you're my idol, Edwin.
Oh, that's very, very sweet of you all.
Gracie, did you say something?
Yeah, I was gonna ask, during your application process, do you ever consider like American medical schools? And because I don't know if there's like any differences between the process between like American and Canadian, like the.
Yeah, no, that's a good question, Gracie. So, very fun fact. Speaking of my life together, getting my life together, right? Very not this situation. I actually gave a webinar to mature students on my application process. I'm like, don't do what I did. To give you a bit of a short timeline, I didn't get into clinical psych. I got rejected around April, May. And then I had a month to like, you know, cry myself to sleep a little bit because it's my second time not getting in. And then I was like, oh crap, I'm finishing up my master's. I have a year, I have no plans to do for the next year. What am I going to do? Apply to med school. That's the next step. So I decided to apply around June. But you may already know, usually people are writing it around that time. So I gave myself about a month to study because I had my thesis and my defense in August. So, you know, very naive and very ignorant of me. I didn't know how much time the MCAT would actually require because I taught undergrad, I taught high school. Like, how easy could this be? And I'm like, oh no, it's actually quite difficult to study for this test, right? And very luckily, I did an all right score. But because of my delay in decision to apply so last minute, American schools, because you were on a rolling basis, it was really not good for me to actually apply because by the time I had my application and my scores, it would have been October. And usually most schools will have their finalized like application list. Even if they say they're open, no one's gonna apply that late usually until like, you know, October, right? So I didn't apply to I didn't apply to the States. So in differences between Canadian and American medical schools, if you're interested in applying to both require the MCAT. American schools are more uniform from what I know and looked into. So meaning that the MCAT is really important for all schools. You have the applications, you have the second wave of application. It's very similar from one school to another school. Canadian, not so much. So U of T, for example, will require the MCAT, but once you made a cutoff, they don't care how high your mark is. They actually care more about you as a holistic application, so they put more emphasis on your essays and your interviews versus your MCAT scores, because I personally, as a teacher, I hate standardized testing. I don't think they actually show how smart you are, especially with multiple choice and on small nuances and details. So, but other schools will care about the MCAT scores in Canada, right? Another thing about Canadian schools, the rate of acceptance is significantly lower than American schools. So about 7 to 8% of application gets accepted into Canadian schools. versus in America, it's about a 20 to 30% of applicants. The side effect to that is once you get into a Canadian school, it's very easy to match. So our matching is about a 97 to 99%, regardless of which school you're in. Versus American, it's very different on your state, your school, your specialty, and especially USMLE. We do not need to write a standardized test after our graduation. So we can get matched without having a USMLE per se. All that they care about is networking. So once you're able to be in your 3rd and 4th year working in the hospitals, all they care about is you passing your exams. Doesn't matter how high your marks are, as long as you have good networking and you'll get into a specialty. So that's the difference. Very hard to get into Canadian schools, but easy to stay and find a job. American schools, a bit easier to get into, but a bit hard to find your maybe competitive specialties.
Right. Yeah. Wow, that's really interesting. Maybe we should invite you back for a Canadian medical school
Yeah,
workshop or something.
yeah, I'm more than happy to help you all out if you want me to come back again or give another talk or something.
Yay.
Yes, please.
Can you practice in America after you get your degree in Canada?
Yes. So the only thing is that you need to then write the USMLE scores.
Oh, okay.
So I have a few friends that are interested in maybe going to the States afterwards, maybe because they have family members or maybe applying to more competitive programs like ophthalmology and certain surgical specialties. So opening up their window for both Canadian and American schools is, something that they should be doing.
Mm-hmm.
The only thing again is that Canadians, we have a bit easier on the USMLE side. We just need to write the test. And the scores are not as heavily affected for us. So again, we get a lot of positive things for being in Canadian school, but it's very, again, hard to get into one to begin with.
Okay.
Mm-hmm.
Wow, thank you.
Yeah, no worries, no worries. You have so much time.
Yeah.
Everyone's probably has said that to you, but I am old, so you all have so much time.
It's just hard when everyone's like,
Yeah,
and then
I can
I
imagine.
want to take my time, but should I be? And
Yeah, I think it's better to take some of, like, if you feel like you're even an ounce a little insecure about your decision, or maybe you feel like you just need mentally a year, that's fine. I know that I wouldn't be where I am with my thought process or my interest in certain things if it wasn't for that gap year. So that gap year really was probably the most important year in my career so far, because it was so like in limbo. I really need to make sure that I knew what I want to get into and not because I was following a status quo per se. Yeah.
Yeah,
Very inspiring.
Thank you. It's very sweet of all.
All right. It seems like nobody has any other questions, but we're definitely going to be inviting you back. You're amazing.
Thank you.
You are definitely fit for education and medicine and like for everything.
Thank you.
Yeah, thank you so much for coming out. I know it's late there. Isn't it like 11?
It's about 11, yeah, 11-ish. I'm a night owl, so I'm most active around this time anyways.
Okay. That's good.
Don't worry about it.
Well,
All right, now thank you so much for having me. Really appreciate it.
Of course.
Thank you so